My name is Frank Dean. I work for Performance Nutrition, a division of Lidochem out of New Jersey. I am the product development manager, and so we're lucky enough to be brought in with this. So we asked ourselves questions, what's going on? There's crops that are affected with disease. I'm going to sort of follow and hit the same things that Don did. But crops are affected with diseases sometimes, and those diseases can't be identified, and they can't be controlled. And so we know that these crops are experiencing physical problems. Crops won't size or ripen. Yields are lower and poor quality. And something doesn't look right, and we know that pesticides and fertilizers don't seem to help. So that brings us into this idea of metabolism. And metabolism is just a set of life-sustaining chemical transformations within all living cells. And it applies to all life, not just plants, but also us. So this all started in northwestern Ohio with tomato growers. I had never heard of citrus greening. I had never really visited citrus before. But what was happening was these tomato growers in Ohio were having problems with bacterial diseases, misshapen fruit, and poor color. The pesticides and fertilizers weren't helping. So they sent me some pictures. And when I saw that, they said that these tomatoes were actually treated with Epiphan, 4 times, and they failed to turn red. So the question I had is, what was it that prevented the pigments from being produced? It had to have happened mid-season because the flower end of the fruit actually had color. So something happened mid-season to prevent the pigments from being produced. So these were just sent to me. So I was looking around, and I saw this. I stole this picture from the USDA. I just want to give them credit. I did not take this picture. I stole it off the internet. But you can see it's a similar type situation with the citrus greening, where the fruit fails to make pigments mid-season. So that's one thing I was thinking of. The second thing is when you look at this holistically, like Don was talking about, and you look at insects and bacteria and fungi and other plant diseases, we see them holistically as nature's garbage collectors. When something is wrong, it's either nutrient deficient or there has to be a metabolic disorder. There's really no other way of looking at it if you look at the world holistically. So these led me to a hypothesis, which is just my own thoughts, that these crops did have resistance to diseases they can no longer control, and that's across the board, whether it's fusarium on wheat or fusarium on tomatoes. These crops had all the fusarium bred out of them, and now they're catching it again. And I believed that the disease symptoms were simply caused by increasing frequency and rate of pesticide applications, accumulation of pesticides in the soil. The pesticides suppressed critical compounds for plant health and their defense systems, and they made changes in the soil microbial ecosystem. So the part of this, another part of this is the changes in cultural practices in the past 20 years. Agriculture as an industry went from very, very labor intensive to chemical reliance initially. It went from chemical reliance to chemical dependency over time. Good farming practices were abandoned in favor of expedients, and years of unrestricted pesticide use have led to a tipping point in what I'm calling indicator crops, meaning that some crops are more susceptible to disease or they are showing the signs of toxicity in the environment overall. So what I said is, in a broad sense, these troubled crops are suffering from metabolic disorders. And a metabolic disorder is any disease that's caused by abnormal chemical reaction in the organism's cells. And when metabolism of a cell, the chemicals are suppressed, they can build up toxic substances inside the organism, or a deficiency of substances needed for normal function, either of which can lead to serious symptoms. We know that glyphosate blocks amino acids. and we look at tryptophan, we know that that's converted to indole 3-acetic acid, which is the rooting hormone. And that gives you cell elongation, cell division, and things like that. We'll talk about that later. Phenyl Alanine is very important. This is blocked. It is a lignin substrate, and it's a natural antibiotic. It is a phytolaxin. It parts per billion. It has the ability to protect the plant. It is the precursor for flavonoids and pigments. Flavonoids are synthesized by the phenylpropanoid metabolic pathway, which is also blocked. And the amino acid phenylalanine is used to produce 4-coumaryl coenzyme A, and that's blocked at the shikimate pathway. Now, what makes that important is this, and this is very exciting to us, is this coumarin-type compounds are actually melanin synthesis inhibitors. And when we look at fungus and we look at citrus, we think about what happened in the past couple of years is this explosion of post-bloom fruit drop. We can tell you that if we supplement the plants with the correct amino acids, we had 0 incidence on our 20 acres in Hayden City of post-bloom fruit drop. And we were lucky enough to have some visitors out there with us to hear the grower talk about it. Tyrosine is very important. It's converted it's just a hydroxy added to a phenylalanine, but it is also a very, very powerful antibiotic and phytolaxin. We all know these are givens. Pesticides are toxic. Some pesticides have half-lives that are very long. They can be stored in the soil and released, and glyphosate blocks the essential amino acids. Like Don was talking about earlier, glyphosate is patented as a biocide. That's much different than saying a bactericide or a fungicide. It is a biocide. It has the ability to kill all life. And it prevents the essential amino acids from being produced. It acts as an antibiotic. It acts as a herbicide. It acts as a chelator. And it selects out, remember, it kills the good guys. It selects out soil microbia that reduces the mineral availability. This is where it blocks it. It's just photographs. I could read it to you, but it's just saying that it interferes with the synthesis of the essential amino acids. This is really the biggest problem is because when we say it selects out these amino acids, that is the primary metabolic pathway. But there's also secondary, tertiary, and quaternary pathways. So when we look downstream in this chemical reaction, if we look at the top right-hand corner, we can see phenolics are blocked. Those are your signal molecules. The phenolics are blocked, so your plant loses any systemic resistance it might acquire or be induced. It becomes impossible without the salicylic acid. It blocks the indole synthesis and indole 3-acetic acid. That prevents you from getting cell elongation. That can give you blooming throughout the year. That can give you all kinds of problems with cell division, no root growth, everything going backwards, tiny leaves. You can't get cell expansion. Then we talk about amino acids, proteins, signal molecules, phytolaxins, all gone. The phenylpropanoid pathway, gone. So there's your antioxidants and your polyphenols, gone. And on the other side, you on the tyrosine side, you lose your pigments, your flavors, your phytolaxins, your pathogen containment. If you don't have lignin, you cannot contain your pathogen. Structural components like suberin, so you can't even hold your cells together, and all your polyaromatics, which then you lose all your antioxidant ability. So this is still my hypothesis, and this is conventional wisdom, that it was caused by a bacterium, vectored by a psyllid. It takes 5 years to show disease symptoms, and the symptoms cannot be induced with bacterial injections. To me, this failed Koch's postulates, so we wanted to investigate that. And the idea that maybe this guy was a wimpy pathogen, like Don had said earlier. Maybe this guy was just an opportunist who had a path into the plant. So what I said is this is my hypothesis before we started. It was actually compartmentalization and concentration of toxic substances made or acquired by the tree, and it shows up as yellow tissue. Now, Dr. Tim Spann wrote at the University of Florida with IFAS that the yellow tissue is the psyllid attractant. So when I read this, I said, what came first, the chicken or the egg? Was the psyllid transmitting the disease, or was the disease there because of the yellow tissue and the psyllid just happened to show up? Deformed leaves, we've all seen it if you're familiar with citrus greening. Abnormal flowers and abnormal flowering events, where you get flowers, the wrong time of year, and pathogen resistance challenges. So this is still my hypothesis. So to test it, we wanted to remove the toxins, apply the essential amino acids and the minerals that are blocked by glyphosate. And the greening symptoms should disappear with new growth. There's nothing you can do for the old stuff. So we proposed that we reduce the applications of glyphosate, begin to understand that bioremediation of the soil is important and the only effective way to lower the concentration of toxins in the soil, and we foliar apply what the toxins block. So we located a grower with 40 acres of dead and dying citrus, all showing signs of citrus greening, and we left 20 acres untreated. Soil recommendation, soil remediation and restoration treatment included a microbial biofertilizer called Nutrismart, and we put that down at 200 pounds per acre. That microbial biofertilizer has the ability to fix nitrogen, dissolve phosphate in the soil, make potash available, and provide and foster beneficial microbes. So it provides shelter and fosters beneficial growth. Then we foliar applied Prudent, which is Prudent citrus, which would be the combination of metals blocked. A little bit different than what Don was talking about. What I did is I took the stability constants of glyphosate with the metal, and I took the negative log of that, which gave me then the dissociation constant. Then I went to Mineral Nutrition Book, and I went to the USDA database, and I found out what minerals are supposed to be there, how much the glyphosate can dissociate, what minerals were actually never going to be let go, and that's where we started. So our mineral mix is a little bit different than other people's. And so that's what we applied, and we also applied a polymer called Argosy in with the herbicide sprays. So this is where we asked the growers to cut back on their chemical fertilizers and replace it with the microbial biofertilizer. We asked them to please reduce their insecticide sprays. Remember I said pesticides block metabolism. Those metabolisms, those pathways, a lot of them are uniform across all life. So we just wanted to reduce the amount of toxins in the field, and we wanted to change the herbicide program. This would be 3 months after we started. The new leaves had no mottled color, clean pointing apex, good flowering position, good size of flower, and the old leaves had the mottled color. I took some samples back to my hotel. The untreated leaves had the death bloom, which you can see. But to me, just being, to me, a flower is nothing more than a differentiated leaf. So to me, that was a hormonal event. What was the hormonal event? Well, you can look at some of the University of Florida's papers, some of their older work, and it tells you that there is an auxin influence on flowering events. But we had good, clean flowers, 3 times, 4 times the size on the apex of the branches. We began to get leaf expansion, 200, 300, 400%. We got no color. We started getting internode spacing. This is Haines City trial. Above the red line is our treated area. Below is a control area where all the trees were pushed. And if I can, this is the grower just talking about it. And if
in Florida, citrus is a $10 billion industry and creates 76,000 jobs statewide. But a condition called citrus greening threatens this critical industry. Greening makes the tree drop fruit before harvest and eventually kills the tree. It's devastating to find citrus greening in your grove. Just ask David Garrett, who's farmed citrus in Haines City, Polk County, Florida, for the past 45 years. When he found greening in his grove, he thought he was out of business. And then he tried the Performance Nutrition Citrus Greening Recovery Program.
These trees were going downhill. They lost their leaves, had a lot of dieback in the tops of the trees. You could see through them. The leaves that they did have were very small, and the fruit got small. The fruit fell off before you would get to pick it. It was just a bad situation. Before we started to use it, the trees had very small leaves. The whole, well, they're falling off now, but the whole tree had leaves about this size if they were that big. You could see through the tree. You could see the trunk on every tree in this grove. And they were just, I thought I'd almost already given up till these started getting the nutritional programs, because all over the world they've been fighting greening with trying everything and had no luck with it. I was as skeptical as they get, and it seems to be making a large difference. All this was last year's fruit. All this on the ground, all these little black things were the fruit that were set on these trees last year that fell off. And this year they're still on the tree. And this is the 6th of February. They're ready to pick. We'll get to pick them.
Waxy, shiny, nice, healthy.
It's a 20-acre trial, and we started treating everything the same way. And it's all I've applied NutriSmart to the soil. I've applied phosphites to the soil. I've applied humic acid to the soil, all performance nutrition products, and all the rest of the products have been sprayed foliarly.
After following the Performance Nutrition Citrus Greening Recovery Program, leaf samples from Garrett's Grove were sent to the lab at Florida State University. The results were impressive. Of the leaves tested from 3 random trees, all but 1 leaf showed little to no presence of the rDNA from the bacterium associated with greening. The grove recovery was confirmed at the DNA level.
The performance nutrition trial started in this block in October in 2013. We've seen a gradual response. Every month or so they look better. The color is improved, the leaf size, they have a lot of normal leaves on them. When we started this in 2013, I'd almost given up. I was just saying, it's land, it's not citrus anymore. And the trees have responded to now where I'm actually becoming optimistic. It's hard to get me optimistic about anything. But if they continue to improve, we may actually have a future with these young trees. The difference I'm seeing this is a more consistent crop. We don't see many of the little bitty fruit now, and it's not falling off. You can't shake this off the tree. You have to pull it. Based on the last 2 years, I think if anybody is serious about staying in the citrus business, they should give the performance nutrition a try. The products seem to be superior to any of the other products that I've used. That's a really good piece of fruit.
So you can't pay an actor enough money to do that, I'll be honest with you. That's David Garrett in Haines City. Okay, so what I did is I went out and I purchased my own citrus trees. I live in South Texas, so it's easy for me to grow them in the backyard. And I began to do my own little test. This is before we did anything with David. So what I was doing is I was placing different compounds on the trees to try and induce the symptoms. And when I got to glyphosate, and I applied a modest amount of glyphosate on a limb, I got the asymmetric chlorosis, which is, in the beginning, what they told us, asymmetric chlorosis was what you had to look for. So I got that with just one dose of glyphosate. The second thing is we get back to this metabolic disorder, and we have to think of it like this. It's any disorder that involves an alteration in the normal production and consumption of carbohydrates, lipids, proteins, plant hormones, and nucleic acids, and they are evidenced by different and various syndromes and disease symptoms. Metabolic disorders produce changes in morphology that includes shape, size, color, and luster of the plants, and it's thought to happen, these are my thoughts, because of a limited pool of amino acids, fatty acids, micronutrients, and a buildup of metabolic precursors in the plant. So the symptoms of the inhibition of amino acids include failure to produce lesions for pathogen containment, chlorosis, which is actually a chlorophyll deficit, another pigment, retarded morphology. Essential amino acids are important for enzyme production, and they also serve as the phytolaxins and lignin substrate, pigments and flavonoids, signal molecules for hormones and the phenolic acids, and they are the structural building blocks of protein and lipid bilayers. So we have oftentimes seen misshapen fruit, and we say, oh, it's oblong. Well, if I had an erector set and I tried to put it together, I can have 1 inch, 2 inch, 3 inch, 4 inch pieces. I can make that erector set look exactly like the picture on the box and operate it just the same. Now, when we think about amino acids, we're going to take another box with another erector set, but I'm going to take all your 1 inch pieces off you and ask you to build it. I'm going to take away some of your amino acids and ask you to build it. You can build that crane. You can probably get it to operate, but it will not look like the picture on the box. And that's the idea behind structural components missing, whether it's suberin, lignins, carbohydrates. So, micronutrients are essential cofactors for enzyme reactions, okay? However, if the enzyme is malformed, as Don was talking about, where the glycine in an enzyme gets replaced with the glyphosate, which they're finding is actually happening. The glyphosate is actually incorporating itself into the proteins, or chemically inhibited, the presence of micronutrients doesn't ensure necessity or use of the plant. So if you have something blocking an enzyme, or you are not able to produce an enzyme like your coumaryl coenzyme A for your phenylpropanoid pathway, if you are not able to produce that, right, all the micronutrients in the world are not going to help if you can't put that protein together or if the protein is folded incorrectly. Again, this is not my work. This would be the U.S. Geological Society. Occurrence and fate of herbicide glyphosate degrades in the atmosphere. Okay, 75% of the rain samples were found to contain glyphosate. All surface water is found to contain glyphosate. It's in the air if it's being used. Here's the problem we have. They say, well, it's 1.5 parts per billion. The rain event cleans it out of the air. Here's my problem with that kind of thought. They're saying, Frank, you go to the doctor, he says, take an aspirin, take 30 milligram aspirin. So a milligram per kilo is a part per million. Now, I take that aspirin and I'm going to help myself with not getting a stroke or a heart attack. It's going to keep my blood thin, prevent it from clotting and things like that. But that's 30 milligrams and I weigh 100 kilos. So we're now talking about a very, very modest amount. Parts per billion aspirin in me has all these biochemical and metabolic changes in my system. If parts per billion can do it to me, why can't parts per billion do it to the tree? Okay? These are treated, fully restored, no greening symptoms, heavy fruit set. This was last year. This grove is restored, no symptoms, 3 years with When we started here, these trees had no harvest for 3 years. The field was abandoned. We only asked to work on people's worst acres. We never asked for when we went there, this was an abandoned acres. There were no harvest here for 3 years. When we go to Hobe Sound, we went to the place that had an out of business sign on their barn and said, can we do work on your land? We did not ask for a productive or a commercial grove. IFAS and the University of Florida say that the PCR reaction is the only way to positively identify citrus greening. So we got Dr. Quee at Florida State University to go ahead and do a primer set to find out which primers we could use for our PCR to identify the pathogen. He found his primer. Then we went back to Haines City. He went to 3 locations, collected samples randomly. And this was taken just this year in January. The trees look great. We went to Haines City and we did the PCR work. And it just shows that we either eliminated it entirely or the titer of the bacteria is so low that it's negligible. And so this would be in Hobe Sound, again, another abandoned grove. And the reason we picked this was the county commissioner said he drives past it on I-95 every day. And he said if we do a test there, he would be able to look at it every day. Well, what happened here this is 5 months after we started. You can see the trees had no leaves at all or very, very few leaves. They begin to get leaves. We're a year into it. They probably got about a 90-95% canopy. We have shade under the tree, beautiful fruit, just awesome. So we went to Dr. Quee visited here. He pulled the samples. We didn't. He went back and did his PCR. And you can see on the sick trees, the bacteria shows up really, really good. See it? On the recovering trees, you can see the titer is way back. On the trees that he called healthy, we eliminated the bacteria 100%. And here goes sick trees. So you can see this is a good farm because it's 35 acres, this field. We treated 15. So we have good controls of very, very sick and very, very healthy trees. In Haines City, we actually went to yield. And with the yield, it is not what you would call commercially acceptable as far as numbers, but it would be his very first yield in 4 years. The fruit quality was very good. The size was good. The sugars, the bricks, everything was good. This is another problem we see in Florida. This is a map from the University of Florida showing greening symptoms and where they occurred in what counties. And when we imposed the map from the U.S. Geological Society of where the glyphosate is being used most heavily, you see that the most heavy concentration of glyphosate overlaps, correlation is not causation, but it's pretty clear. This is how we reduced another video. This is how we reduced our herbicide applications.
This grove where we're standing now has every weed known to man and probably 4 or 5 that haven't been identified yet. It's good sand, good as Florida soils go, it's good sand. And it grows the heck out of weeds. The only weeds that you see in here now are these winter weeds. There's all the ragweed and the grass haven't grown back. I treated it, herbicided this grove twice last year as opposed to like 4 or 5 times before. And that's just adding argosy to my glyphosate and my landmaster. And I cut the rate of my glyphosate in half with the addition of argosy. Argosy is a polymer, and it's not really, it is a surfactant that makes it spread, but it cuts the leaf evenly, the weed evenly with it, and it takes longer for the weeds to die, but they die almost completely. You don't have many escapes.
Argosy RF from Performance Nutrition is an environmentally safe polymer adjuvant designed to keep chemical applications on the plant and weed longer. Argosy RF helps to reduce the rate of chemical pesticides and increases the interval between sprayings. This saves you time and money. How it does this is it's rain fast and it's elastic. Argosy RF keeps the active ingredient on the target, even through irrigation and rain events. It also stretches as the plants or weeds grow, and every time it's moistened, it redistributes active ingredients on the target, including the new growth. Argosy RF is completely inert, so it's safe for the applicator and safe for the environment. Argosy RF helps reduce pesticide runoff.
I've tried Argosy in the copper spray in a couple groves, and last summer was very, very, very rainy. And you spray the Argosy, and a month later you could still see copper, the coastline residue on the trees.
So we went from 5 applications of glyphosate down to 2, right? So that's a 60% reduction. Then he cut his glyphosate in half again. So now we're 80%, which also eliminated 3 applications of the Landmaster. And Landmaster is actually a combination product of 2,4-D and glyphosate blended together. So you can see glyphosate, these are just my notes, glyphosate inhibits the production of amino acids. That produces pathogen resistance failures, prevents inherent, systemic, and acquired resistance, inhibits enzyme production, blocks plant hormone production, and it inhibits pigment production. Soil remediation becomes necessary for reducing the total toxic load. Foliar application of what the toxins block or what's inhibited includes the amino acids and the metals. And we have to lower the total toxic level or the toxic footprint by reducing or eliminating the use of glyphosate that inhibits the plant shikimate pathway. So with that, I'm going to stop right here. If anybody has any questions, you're certainly welcome to ask. Yes.
If I could, I'd like to make a few comments.
Yes.
I strongly encourage you to actually conduct replicating scientific trials to demonstrate the efficacy of these products.
Right.
These observations are a good start.
Right, exactly right.
Actual scientific trials.
Yes.
Extremely useful.
Yes.
I also just want to point out that you can get symptoms of HLB without applying any pesticide. We routinely infect trees in the greenhouse as part of our research program.
Right.
And you can get all the same HLB symptoms.
Right.
And just one other fairly minor point.
Yeah.
A milligram per kilogram is a part per million.
Right.
Part per billion.
Exactly right. But I weigh 100 kilos. So take that part per million and divide it by 100. You're in the billions. It's parts per billion. I weigh 100 kilos. Right.
If you have a part per billion or something in your 100 kilos, that is
It's a very modest amount.
it's a very tiny amount.
Right.
It's not the amount that
No, I said I took 30 milligrams,
yeah.
right? So that's 30 parts per million per kilo. I divide that by 100. I weigh 100 kilos. I am now 0.3 parts per million, right? Or 300 parts per billion. So we're in the parts per billion. It's pretty simple math. Yeah.
I do encourage you to actually conduct experiments.
Oh, we would certainly, we would love to. We would love to. We just can't find anybody who's willing to work with us.
Well,
Yes.
just as you spend money in developing products,
Yes.
this is part of your product development.
We know that. We know that. We truly do. Go ahead. Yes, ma'am.
So I just want to reiterate what was said in regards to the trial for grid observations. How do you explain how given your glyphosate theory is kind of like the green organic growth as well?
Well, when you look at, there's, I don't want to bring organic growers into this, but when we look at their irrigation water, it certainly contains parts per billion glyphosate. In reclaimed water, after every irrigation event, that concentration of glyphosate in the reclaimed water actually goes up. There is no organic orange juice that does not have glyphosate in it, okay? It's in the rain. But more importantly, what do they use to fertilize? They use manures. What do we feed our animals? We feed them GMO corn, beans, soy, GMO alfalfa. They are essentially like we are. We are just fermentation units. We are mobile fermentation units. We eat the food. What our body doesn't need, it concentrates and disposes of. That's what's happening, say, with chicken manure and cattle manure, is they eat the corn, they acquire the proteins and the carbohydrates, and their waste actually begins to concentrate the glyphosate. That's what we're putting down under the trees. You can send it into the lab. I've seen a ton of chicken manure have the equivalent glyphosate as a labeled rate of glyphosate. You've seen that, Don? Yeah. So it's common knowledge. There is no rule in organic farming that organic herds or fowl be forbidden to eat non-GMO food. There is no rule. So all organic essentially is being contaminated with the same problem. It's just in manure it becomes a slow release source of glyphosate. Yes, sir.
If I understand you correctly, you saying that glyphosate substituted glycine is working?
Yes, we can see that work coming out of MIT with Stephanie Senoff and Samsel. They have some papers. I believe one has been accepted, peer-reviewed, accepted, and published, and I believe they have some more. And the concern there is that in proteins, and there's proteins in your bones actually, you can find glyphosate substituting for glycine in protein. It's at least that's the way I read the papers. Is that how you read them, Don? Yeah. That's exactly how we read it. And what that does is it malforms enzymes. Because the glycine and the glyphosate have different shapes, the enzymes become malformed. And when they become malformed, they no longer function as a catalyst, as a biological catalyst. But that's true. There's going to be 3 or 4 papers coming out, I'm sure. What journals? It's out already, right? Yeah, I don't. Yeah, it's probably European phytopathology or something. Yeah. Yes. I hope so.
Thank you.
Any other questions? I was hoping for more questions. Anyway, to give you an understanding of what's going on, this is a death bloom that I induced, and we got it 45 days after application. That's just something I did. So that bloom came 5 months too early. This explains the tiny fruit. This is a soybean field that was accidentally sprayed with glyphosate. So we do the same math, get the USDA numbers, calculate the dissociation constant, and apply what the glyphosate blocks with the minerals, and we can stop the glyphosate from working. This would be something I just wanted to share with you. They say that it locks up and binds up in the soil. This is pumpkins in Ohio. These leaves had 237 parts per billion glyphosate in them. There was no glyphosate applied to this field for over a year. This just shows that we did replicated studies showing that we have the organisms that do actually consume and metabolize it. As far as what the university recommends, this here was a paper put out saying that glyphosate is safe for citrus. One application made to brand new plants. The interesting thing here about is they only used 32% of the labeled rate in their study, and the plants were hand weeded for 2 years. It took 8 months to 2 years for the trees to recover from their first application of glyphosate. However, in Florida, we do 5 a year. So you can see where this roller coaster is going. This is actually a USDA Canadian plant. This is the amount that the university recommends of glyphosate. They're saying, you know, let's put 3.5 to 4 pounds per acre of active ingredient per spray 5 times a year. That is 20 to 25 pounds of active ingredient sprayed directly under the drip line. Everything else is mowed. So you're talking about 40% of the acre getting 20 to 25 pounds of glyphosate per year. That is insanity. So, anyway, I will,
I have a question real fast.
yeah.
So how long are you going to follow this grower? Are you going to keep with him, seeing if his yields are...
Yeah.
And that was this video taken last?
No, that video was taken in January of this year.
Okay.
Or February 2nd. Yeah. Just my references and stuff, that's all I had in there. Everything was documented. That's our story, and we're sticking to it.